Good morning, good afternoon, good evening, and I know good night to some of you because those of you who are coming in from Australia, it's definitely nighttime, and we salute you in terms of participating with us. Those of you who are on the west coast of North America, we also salute you for getting up to do this. So we're really right across the globe in terms of the participation in this workshop. My name is Durham Wong-Rieger. I am chair of Rare Diseases International Council, and I'm also the president and CEO of the Canadian Organization for Rare Disorders. And I am so really delighted to be able to welcome all of you to the second in our series on advanced therapeutic products, or also known as cellular and gene therapies. And we are just really pleased in terms of the participation and the interest that's been shown. I wish we had time for everybody to introduce themselves, because I think you'll find it, really enlightening in terms of what we do. What we're going to do is we will have two panel discussions, and we'll have a Q&A after each panel. So you may, in fact, throughout the presentations, please do use the chat box to put in your questions. Feel free also to chat in the chat box. We'd love to have it if you could introduce yourselves in the chat box, indicate what country or what group you're coming from, so we end up with a nice running commentary on it. Those of you who are unfamiliar, well, everybody's familiar with Zoom by now. If anybody's on here is not familiar with Zoom, I think you've been living under a rock for the last year, so we won't even bother trying to explain to people how you use it. But we do not have the opportunity to have much of a live Q&A during the panels, but do put your questions into the chat box, and we will try to raise them with the panelists as we go on. The workshop is actually divided into three sections. The first one will be an introduction to genome editing, and really the science behind it builds on what hopefully some of you will remember from the first session on gene replacement therapy. The second panel will really talk about genome editing and some of the issues involved in it, including some of the issues that have been raised in terms of the applications around germline genome editing. But that will also then be the feature of our third session, where we will have you in breakout groups. We're going to introduce a question that we would like for you to discuss in your panel. You'll see it's sort of a simulation around a real issue, and we're really, really delighted to have all of you on here. The only other person I'm not seeing on the moment on my panel is Nicola Walsford. So if we'll get to that hopefully before we get done. So hello to everybody who's here, and because we have such a packed agenda, I'm actually going to open right up in terms of the beginning. So if we can just put the workshop slides up, I think that'll give us an introduction to the session. There we go. All right. There we go. All right. So these then are the overall objectives. We'll just move on. Hopefully everybody's kind of gotten into that already. We really are, this educational sessions, want to have the opportunity to have some interactive discussions. Hopefully many of you will have had a chance to look at some of the materials. They remain available to you. So these are the basic issues, and you'll see the last part we talked about in terms of some of the ethical issues around genome editing. This is what I just said to you, the two presentations. Our presenter in the first panel will be Greg Davis. He's a chemical engineer, he says, now working at Sangamo Therapeutics, and has always, as he says, worked very much in biotech. We've invited Christine Moberg, who's also from Sangamo, who I have known for a number of years, and I'm just such an admirer in terms of the kind of educational material she's been able to put together. Delighted she can join us. And Nicola Walsford from Jesse's Journal, we're going to ask to make some commentary around it from the point of view of a patient organization, and certainly patients and parents that are looking at genome editing as a possible therapeutic area. The second panel actually will be, one slide back, is going to be kicked off by Ron Perlman, who is I guess he's Professor Emeritus now at York University, who's going to give us some overview again in terms of picking up where Greg talked off in terms of genome editing. Nick Mead, who's with the Genetic Alliance UK, our sister organization in the UK, and Ian Stedman, who is a colleague, a parent, a patient, and also then a professor at York University with a degree in law and in PhD in, I think, in public health. So delighted to have all of you here. And I think next slide will just sort of show you kind of where the rest of it's going to go. So this is the exercise we'll have. We'll come back to it at the end. So I think without much further ado, I'm going to ask Greg to step up and share his screen and take us into the exciting and challenging world of genome editing.
Okay, thanks, Duran. Let me share my screen here. Can everybody see my screen okay?
Yeah, I think you're good.
Is all good? Okay. All right. Well, thanks very much. It's a pleasure to be here. Good morning, good evening to everybody. I know you're from all over the globe here, so all times of day. My background is in genome editing, so I work at currently at Sangamo Therapeutics, but I've spent almost 20 years now working in the genome editing field, primarily working on the tools used to access and edit the genome. And so in this session here, what I really want to do is give you an idea of what the genome is and some idea about the tools that we use to edit it. And I'll walk through an example later on for one particular disease to kind of show for this particular disease how there's a variety of ways you can address it. And it's a good illustration of different approaches that you can use. And then lastly, I'll talk at the very end about genome editing versus gene therapy. So a lot of these terms have a broad meaning, and I'm hoping towards the end of that to give you some sort of a distinction between what we talk about with genome editing and gene therapy. So yeah, so and I can see everybody putting their name in the chat there. So yes, please keep doing that. It's great to see where everybody's from. So I'll just start off with a quick quiz here. So I don't know if all of you can see this, but this is related to the topic I'm going to talk about. Can anybody type into the chat what they think this is an image of? Everybody just take a little bit of time and type into the chat what you think these might be. So I see someone saying, Sarah saying DNA, Dana saying socks, genes, chromosomes. That's right, they look like chromosomes, but they're actually socks. So this is some art by Gina Glover. She's an interesting person that does that takes biological images or biological themes and works them into art. And so she took a look at chromosomes and thought, wow, they look like socks. And so she made this great display of it. And this is displayed in Guy's Hospital in London. And so I'm a visual person, and so I thought I would kind of use that image of the human genome to sort of orient how we talk about genome editing and how we access it and work with it. And so here's an actual image of chromosomes. And if you look here, you can see there's 2 copies of every chromosome here. They're ordered from 1 to 22. So if you look at them, going progressively in these increasing numbers, the way they line them up this way is because of the length of them. So chromosome 1 has the most DNA in it, and chromosome 22 is the shortest. And if you add all your DNA up in the genome, there's about 3 billion letters spelled out in A, C, G, and T. And so it's a lot of information packed into your genome. And oh, I see someone in the waiting room here. Let me, okay, I think that was taken care of. Okay. So there's within these 3 billion letters, there's about 20,000 protein coding genes in there. And these genes are small parts of the genome, each that have a unique function. So they all have very unique functions for all the processes that they need for the cell to survive. And so this expanded view of the genome here showing you sort of a bird's eye view of all the chromosomes. If you started to zoom in a little bit closer, if you look on the lower right hand side here, you can kind of see a closer up view of the chromosomes, and you start to see some fine texture there about how all the DNA is packed up and wrapped up. So this DNA is wrapped up very tightly, and if you zoomed in any closer, you would start to see all the bases of A, C, G, and T there that would spell out all the letters of the genome. Okay, so so the next question is, where is the genome? So within most cells in your body have a copy of the genome. So the one example of an exception are your red blood cells. So your red blood cells are enucleated. They don't have a nuclear copy, but the vast majority of cells in your body do have a copy, and they have certain genes turned on to support the functions of wherever those cells are in the body. So within those cells, there's typically 2 copies of the genome per cell. Each person has trillions of cells in their body. So as you can imagine, what that means is you have trillions of copies of the genome throughout your body, all functioning in different ways to support the functions in different tissues. So, and each cell is expressing a different part of the genome. So I mentioned that there are 20,000 different genes in the genome. And basically, there's, in each cell, there's a different sort of collection of those genes that are on and some are off to support the functions of those particular cells. So what is a gene in particular, and how does it cause disease? And looking at this, this image of the chromosomes here, let's just zoom in on chromosome 11 here, for example. And so what I'm going to use as an example to describe how we can use genome editing tools to address diseases, I'm going to use sickle cell anemia. So the gene for sickle cell anemia is hemoglobin beta. It's located on the P arm of chromosome 11 here. So I've put an approximate location with these yellow dots here about where that gene is on chromosome 11. Chromosome 11 is about 135 million bases. So again, remembering that the entire genome is about 3 billion, still pretty large amount of information here. The gene for hemoglobin beta is about 1,600 bases. Okay. And so if we zoom in a little bit closer and get down to the point of the disease here, what you're looking at here on the right hand side is the actual genetic sequence in A's, C's, G's, and T's. And if you look very closely here, you'll see a T that I've shown in red here. And so that is the site of the sickle cell mutation. Okay, and it's that one change in that one base. Again, 130 million bases in total on chromosome 11. And here we're talking about this one base in hemoglobin beta that is the source of the mutation. So if we look at the normal, sorry, I have a dog here wanting to get up. So if we look at the normal sequence, which would code for healthy red blood cells, you'll see that this is the sequence is an A here. When it's mutated in the disease, you'll see that there's a T at this position here. And so what that does is that causes a change in the amino acid sequence, and that will cause this glutamine here to be changed to a valine, and that will change the structure of hemoglobin. And eventually what happens is it causes the red blood cell to change shape, and then whenever that flows through blood cells, it sometimes causes the blood cells to sort of clog up in blood vessels and cause vascular occlusion events, which can be very painful for sickle cell patients. Okay, so how do we correct this misspelled gene here? So with it being one letter, just a T, the inspiration once that was known by scientists was, how do we access that? How can we access this chromosome and go in there and edit it like text to change that T back to an A? Okay. So there's in 2005, there was a publication that came out on zinc finger nucleases, and basically what they showed was that you could edit the human genome and human cells. And prior to that point in 2005, there was only you could only edit the genes. And hang on just a second here. I'm going to move my dog into her pen. I'll be right back. Okay.
So this is a good opportunity if people would like to enter some questions into it. And just to give Greg a break, Greg is on the West Coast of the US. So it is 5 o'clock in the morning for him. So we can give him a little break in terms of needing to do something with his doggy and his willingness to come on very early in the morning.
Yes, I'm back. Sorry. Yeah, I have a dog. Her schedule's off. So yeah, I was up at 5 a.m. So she's trying to figure out what time of day it is. So sorry about that, Duran. Yeah. So if we go back, if I go back one slide here, yeah. So, changing this one T back to an A. If we go back to 2005, yeah, there was a great publication that came out showing that you could edit these chromosomes in human cells. And before that time in 2005, it's just that scientists couldn't access the chromosome. They couldn't get in there and edit it in human cells. And prior to that, the scientists had been able to modify mouse genes and change genes in mice and see what the effects of those mutations were in mice, but could not do that in human cells. And so these tools that you've probably heard about are zinc finger nucleases, TALENs, and CRISPR. So these are all genome editing tools that you've probably heard about in the press. They all bind genomic DNA. So one core function to each of these is that you can change them and tell them how to bind DNA. Some differences between them are that zinc fingers are from mammals, and CRISPRs and TALs are derived from bacteria. And when I say zinc fingers, CRISPRs, and TALs, these are the DNA binding portions of these molecules, okay? And so it's very important. They're kind of like the find function for the human genome. So when I showed you all that text of the G, Cs, As, and Ts, we can use these to go in and find a particular sequence. And so the inspiration then with these was that we can program them to go find the sequence around the sickle cell mutation and possibly change it. So all of these can be edited to edit the genome. And it's great that we have all these tools because different genetic diseases require different tools. So when we go outside sickle cell anemia to other various locations in the genome and different diseases, the genetic context is different in all of these, and genes function in a huge variety of different ways. And so it's really great that we have all these different tools to address all these different diseases. So, okay. Just to give you a little bit more detail about zinc fingers. So again, they can read, grab, and edit the genome. So I'm showing in red here, if you look at this zinc finger, it's showing you in a little bit more detail how the protein's interacting with the DNA. So it's kind of showing you how the protein can see the DNA. So it's actually seeing the G, Cs, As, and Ts there. And so we can change these zinc finger proteins to see certain sequences. And the analogy there is like if you went into a library and you wanted to pull a particular book off the shelf, that's kind of what a zinc finger or a TAL or a CRISPR can do. They can pull that off. And then once you've identified that particular sequence, you want to edit it. So we can attach things to these to zinc fingers, TALENs, and CRISPR to cut the DNA. Do all sorts of things. So I'm going to talk about editing today, the sort of the scissor function, which is cutting the DNA, very much like you would cut a strand of yarn. But there's a lot of other things you can do with them. So the zinc finger, again, in this case, I'm showing you zinc fingers, those portions just bind the DNA. There's many other things we can attach to those zinc fingers and do different functions. There's things called nucleases, which are scissors, like scissors that cut the DNA. But there's other things that we can attach, which this one over here that's called CRAB, K-R-A-B. This one can kind of turn a gene down, not necessarily knock it out completely, but make it function quieter. Okay, so if there's a mutation in a particular gene and we want to just quiet it down to a healthy state, that would help do that. On the other hand, if there's a gene that's not expressing as much as it's supposed to, we could activate a gene. So in the upper left-hand corner, P65 here, so we could actually go and find a gene and turn it on. Okay, but what I'm primarily going to talk about today is this sort of scissor editing function where you can edit edit the genome like text. So I'm seeing some yeah, so if you have any questions, please put them in the chat. So so everybody, someone's asking what the name of my dog was. My dog's name is Faye. So yeah, thank you. Yeah, if anybody has any questions, just put those in there and we'll run through. I'm going to move through this in the interest of time, and then we'll try to scroll back through these and answer those at the end. Okay. So for sickle cell, let's go back to this T here. And let's say if we wanted to correct this T back to an A, let's say if we wanted to cut it with a zinc finger and paste in a new sequence to make it back to an A. So scientists have been able to do that. So in the lab, if they take human cells, they've been able to take human cells in a dish, cut this location and change that back to an A. And it seems to work well in cells in a dish in the lab. But when they try to translate this method in the clinic for patients, it doesn't seem to work as well. It's just not efficient enough to result in a sequence. Okay, to result in a therapy that's very effective for patients, effective enough to deploy to multiple patients. And so what that sort of made everybody go back to the drawing board and think of, you know, what can we do then? What's really the best treatment for sickle cell? And another idea is to, instead of trying to correct this, you know, this incorrect gene on chromosome 11, hemoglobin beta, what if we just put in a whole new copy of hemoglobin beta? And to use the text analogy again, it's almost like pasting in a whole new paragraph into a book. So let's just paste in a completely new but correct copy somewhere else in the genome and have it be expressed. And so this is essentially, this is the concept of gene therapy in a sense. So there's, we're just pasting in a whole new copy of that gene instead of editing the native one, okay? And in this case, they're using a virus called lentivirus to place in a whole new copy of hemoglobin beta. There's a company called Bluebird Bio that's treated patients using this approach. And I believe they have some amount of approval in Europe and are still working on this in the US. And so this is this lentiviral approach, this gene therapy is an effective approach for sickle cell anemia. So as a third example, I'll talk about one way that the company I work for, Sangamo, is collaborating with Sanofi to develop. And this is going back to editing again. So we're using the scissor function of what we call ZFNs here to cut on chromosome 2. And when we cut on chromosome 2, instead of knocking the gene out or changing it in a specific way or correcting it, when we cut at this particular location, it has this amazing result of turning on fetal hemoglobin. So when you're a baby, there's a fetal hemoglobin that's on, but as you grow into an adult, this fetal hemoglobin gets turned off, and you have other hemoglobin genes that come on as you get older, and take care of most of the functions of grabbing oxygen and ferrying that over to your lung cells, ferrying that from your lung cells to every cell in your body. And so in this amazing sort of bit of biology here, scientists figured out that there's a place we can cut in the genome and turn fetal hemoglobin back on. And so it's a very simple and effective approach. And so basically we design zinc finger nucleases to go cut on chromosome 2, and this turns on fetal hemoglobin, and that can compensate for the adult hemoglobin and start to reduce the number of vascular occlusion events there. And so again, Sangamo has been partnering with a company called Sanofi to deploy this treatment in the clinic. And so this just kind of shows you, you know, this is an amazing example of the importance of having really diverse and wide funding in medical science to know all these details about all these different genes, because again, when scientists first found out about how this one base was incorrect in sickle cell anemia, it's the obvious thing is to correct that one base, but because of all the scientific information, all the medical biology that's out there in the literature that's supported by various countries and basic science institutions, we knew about fetal hemoglobin, the genetics behind it, and how we could use zinc finger nucleases to go in and cut that particular location. Okay, so this is technically what I would call a genome editing approach. So we are cutting on chromosome 2 and turning on a fetal hemoglobin somewhere else in the genome. So how this works, this third example, how this would work in the clinic, just to give you a brief, a very quick overview of how that's deployed, is blood cells will be taken out of the patient, apheresis, so in step 1 here, and then those cells would be taken and exposed to the zinc finger nucleases outside the body. And then the zinc finger nucleases would go cut on chromosome 2 here, like I showed you, and this is near a gene called BCL11A. And then once that region is, once the zinc finger nucleases have cut, those cells are harvested and then infused back into the body. And by that time, they've started to express fetal hemoglobin, and then those can go back in and compensate for the loss of adult hemoglobin. So this is what you would call an ex vivo therapy, where cells are taken out of the body, edited with the nucleases outside the body, and then reinfused back in. Okay. And that's different from an in vivo therapy, where genes are put directly into the body and then have to go to various locations. And so that's kind of where I wanted to end up here in the last 3 slides, is to talk about, what's next for genomic medicines, and what are the major differences here between gene editing and gene therapy. So to do in vivo editing, to get genes or zinc finger nucleases or TALs or CRISPR into the body in various locations, we have to have a way to tell them where to go, which cells to get to. Do they need to go to the liver? Do they need to stay in the blood, go to the brain, et cetera? And so I'm using here a message in a bottle sort of approach, because if you release a message into the ocean, it's floating around seemingly haphazardly. And so the analogy here is, if you put it in, a genome editing reagent into your blood, it has to go through the circulation of your body and then wind up where you want it to. And so there are that's a scientific challenge to do that, to get these molecules to go to the exact place that you need in the body. One amazing technology that's out there that you may have already heard about in your training here with Cord is adeno-associated virus, or AAV. And so this is an amazing technology that's been developed in the last few decades. And what they can do is basically change proteins on the surface of this virus and tell it which tissue to go to. So they can program it to go to skeletal muscle, the heart, the liver, the eye. So there's a lot of different places this can be ferried to. And the function for AAV, it's very similar to what I mentioned for lentivirus before, and this one example of sickle cell anemia, where Bluebird is taking a new copy of hemoglobin beta and sticking a whole new copy of that gene in the genome. So adeno-associated virus does that same approach as well. They're taking a whole a corrected copy of an entire gene and bringing that into a cell. And the excellent part of AAV is that we can target that to various tissues. And so this is a virus. I know for some people, virus has a negative connotation these days, but there's been enough work going on with biology that many of these viruses, they've been changed. And in particular, AAV, this one, it's not known to cause a disease in nature, and scientists have changed it and rearranged it so that it can function as a safe way to deliver corrected genes. And so some of the diseases that it can be addressed with it are hemophilia, PKU, SMA, Fabry disease. These are all diseases that we can take a corrected version of the gene, package it into AAV, and deliver it into the body. So one big question here is how AAV and lentivirus are different. So both of them deliver corrected copies of genes to the cells. Lentivirus inserts corrected copies into the genome. So in a way, lentivirus, even though it's a gene therapy, it's kind of like a form of genome editing. It is actually putting, stitching a corrected copy back into the genome, whereas AAV maintains that genetic copy outside the cell. So it's kind of floating around almost like a satellite there and expressing that new copy away from the chromosomes and not necessarily stitching it into the chromosome like lenti. But they both have the general function of putting a new gene in and are therefore sort of technically classified as gene therapies. And so this is my last slide here, and I'm hoping this is just really driving home the difference between these two general terms here. In genome editing, what we're talking about is changing the sequence of your endogenous chromosomes, either with zinc fingers or CRISPR. And with gene therapy, we're primarily talking about putting new copies within the cells and bringing those in for various therapies. So with that, I'll stop here and take some questions. So
Yeah, there's been a good number of questions. Thank you so much, Greg. I wonder, in the interest of time, I'm just going to introduce Nicola Worsford. Nicola, I don't know if you can turn on your camera or somebody, and who is in Canada with us. She's well, she can introduce herself. She's with Jesse's Journal. I wanted her to comment just briefly on terms of what she's heard as a parent of a child with Duchenne muscular dystrophy, what this might mean to her, and what the anticipations might be, both in terms of gene editing or in terms of genome editing or gene therapy. Nicola, are you there?
I'm here. Hi, everyone. Good morning, good afternoon, and thank you, Durhan, for inviting me to be part of this panel today and sharing our experience with Duchenne muscular dystrophy. As Durhan mentioned, I work with Jesse's Journey. I'm a director of research and advocacy, but also a mother of a son who has Duchenne muscular dystrophy. And this is a really exciting time for our families. And CRISPR is definitely, CRISPR-Cas9 is one particular technique of genome editing that's been in the news quite a bit with our community. There's a number of researchers that are currently working on this, some of which our organization, Jesse's Journey, are supporting through research grants. And we have a lot of optimism about this. Duchenne muscular dystrophy, it's caused by mutations in the dystrophin gene. Which is essentially a gene that acts as a shock absorber that allows the muscles to contract and relax without being damaged. When the symptoms of the condition is that it starts in early childhood and it renders our young children and teenagers in a wheelchair, and which eventually they do pass away from in adulthood or young adulthood due to respiratory and heart failure. So there's a huge unmet need to find effective treatments for Duchenne. There are many therapies that are under investigation right now, which is exciting for us. And a lot of these approaches are looking to find ways to correct the damaged dystrophin gene, but they do all fall somewhat short. Some of them at most are able to certainly delay the progression of the disease. But a lot of these aren't, so for example, a lot of these are able to produce either a truncated version of the protein or aren't necessarily repairing the actual DNA and replacing it with a full functioning gene. Gene therapy is a really exciting area for us right now as well. But because this dystrophin gene is so large, it can't fit into these AA vectors that we were just speaking about. And so scientists have been able to fit in these sort of mini dystrophin or micro dystrophins into these gene therapies. But again, this is just replacing, it's essentially replacing dystrophin. It's not correcting. So CRISPR-Cas9, one of the techniques that's currently being investigated, is really exciting for us because it has the potential to permanently repair that mutated gene and restore that dystrophin gene to its full potential. So it's able to produce that fully functional dystrophin protein that is lacking in Duchenne families. So ultimately, this is the cure that our community have been waiting for, and that's why it's so exciting. But we do need to balance that also with the expectation. I don't know if you've been keeping track of the news, but there are a number of researchers, some of them right here in Toronto. Dr. Cohen and his lab, for example, are working on the CRISPR-Cas9 techniques to be able to fix what's called duplications in this dystrophin gene. And they have had success in enabling to do this in mouse models. But again, because it works in the mouse model doesn't necessarily mean it's going to translate into humans. And so this is an area that is still in its infancy. It's still in the early stages. And what's paramount to our community, obviously, is the safety. We want to make sure that and it is essentially precision medicine, because it has the potential to go into the specific gene, change the mutation or the damaged part of that gene and replace it with the right piece, producing that fully functional gene. But we want to make sure that it works properly, that it's changing the right gene that it's supposed to be, or sorry, changing the right part of the gene that it's supposed to be, and that it's not, it's not having other effects in other parts of the body that could damage it. And I always use that analogy of the house. And if you imagine, if you're renovating your home and you want to take down some walls to open it up so that it flows and functions better, you only want to take down those beams that aren't structurally keeping the house up. Because if you take down those structural beams, then the whole house is going to fall down. So we want to make sure it's only targeting those walls that are going to improve the function of dystrophin.
Okay, so thank you very much. I wanted to make sure that we have a sort of a practical application as well, but maybe I'll turn it back to both Greg and Christine quickly. I know we've gotten lots and lots of questions there, and maybe this kind of leads into that. I think you talked about it a bit, Greg, as well. One of the questions around why does it seem to work better for like beta thalassemia than for sickle cell disease, or what are, listening to what Nicola is saying as well, where would genome editing be most useful as opposed to, say, a gene replacement therapy? And then her notions of what are the big risks in terms of a genome editing?
Yeah, so in terms of where one would be better than the other, a good example is in the case of sickle cell, whenever they wanted to correct that one single base, one limitation there, if you want to get down to the real technical limitation, is to edit the genome like text, there's a process called homologous recombination, where you take the sequence and sort of stitch it. It gets copied into the genome, and that's the more technical term for that is DNA repair. There's a whole field of that studies that. And in your body, different cells, they don't all do DNA repair the same way. So especially non-dividing cells. So let's say like brain cells, if they're not dividing, we can't do this sort of text-like editing. So, so since you can't do that, you have to develop, you have to come up with a different way. And so, in that case, they came up with these alternate methods for, turning on fetal hemoglobin, or using lentivirus to put in a good copy. That's just a good example for that one particular disease of deploying various modes of genome editing and gene therapy.
Good question here. Says, are these therapies long lasting? Do they have to be repeated, or even more importantly, can they be repeated?
For genome editing, the hope is that if you hit enough cells in the body, that those cells would, if you modify the chromosomes, when the cells replicate, they carry that mutation with them in your body and then is more of a permanent fix. That's the hope for the genome editing approach. Okay, you know, so for the adeno-associated virus approach, it is very it's very durable. So it can replicate. It can stick around for quite a while and give years of benefit. But the important thing to note with AAV there is it's episomal. It's not inside. It doesn't get stitched into the chromosomes. So that's one of the major differences between those two. So.
So we will have our third workshop, actually, for those who are interested. We're really going to focus in on the lentivirals, and we're actually going to, somebody asked the question about cystinosis. I think cystinosis and some of the LSDs we're going to be using as an example. So this is a brief commercial for the third workshop coming up. If you're interested in that question, this is what we're going to be doing. We're really totally out of time, but I wonder if I can get Christine who's been so kind in terms of helping put this together, just to give us a brief update in terms of how is Sangamo using these genome editing, and what are some of the disease areas that you're looking at? I know Greg mentioned it briefly.
Yeah, no, thanks so much. I'm happy to jump in here real quick. So, I think this conversation is wonderful because the differences between the approaches is really important for the patients and the communities to understand. So a lot of the work we're doing is really to help educate on the various approaches for genomic medicine. Sangamo is unique because we have multiple approaches. So we do gene therapy. We have gene editing both in vivo and ex vivo, as Greg mentioned, as well as gene regulation. And so currently we're working in the areas of hemophilia A, beta thalassemia, Fabry disease, PKU, and we also have CNS related programs related to tauopathy. So there's multiple approaches that can be used, and just making sure you're using the right approach for the right disease and for the patient is really what we're focused on. And the more educated the patients are, the better it is, so they can have conversations with their healthcare professionals. So thanks for the opportunity, and really a great session.
Yeah, I really apologize for not being able to make this the full session. If we had kind of known this, we could have done the entire hour and 2 hours just on this, just on the questions that are coming up. I definitely, we have planned this as a 3 workshop series, but as we've been talking about it, we think we have lots of opportunities for having much more, maybe as people become more educated as well, some real just discussion sessions as well. So my huge thanks to Greg, huge thanks to Christine, and huge thanks to Nicola, who, as one can see, is certainly what happens when you've got an educated, intelligent patient-parent community that can really, really help to direct some of that research. So huge thanks to all of you for your participation, and look forward to it. I think, Greg, you're going to stick around with us.
Yeah.
So we can
I'll hang around.
have Greg in some of the breakout sessions as well. So thank you so very much. Okay, we are going to make a quick shift into our second panel, which is going to be picking up a bit on where Greg started off. I think you'll hear that right away, and really starting to move into some of the issues around genome editing, the uses of it, and some of the controversies that might be ensuing. So we've looked at the uses of it in terms of therapy. I'm going to turn it over to, they're not entirely just academics, but they are going to provide us with a somewhat of a learned approach in terms of the issues around genome editing. So we've got 3 presenters here. I think I introduced them briefly up front, and I'm going to turn it over directly to Ron in the interest of time from York University, who's going to start us off with a bit of an extension around Greg's talk in terms of genome editing, and then we'll get into some of the issues around it. So Ron, can I turn it over to you, please?
Yes, can you hear me, hopefully?
We got you.
Okay. So thank you very much, and welcome everybody, whatever time of day or night we happen to be in your area. And I am going to follow up from this wonderful introduction, and there's going to be a significant amount of repetition, as you'll see, but repetition to those of us who are academics is good. It's the way we learn. And much of what I'm going to focus on is from the 30,000 foot or 10,000 meter, depending on where you are in the world level and just present some things from an overview because of time. So let's just get right into it. So you've heard about genome editing. Genome editing is important in all of the various technologies that we've heard about and that I will mention as well. I'll focus on CRISPR because it's the one that is presently being very much utilized and has such great potential. And some of the advantages of the CRISPR kind of approach is that it is programmable, it's specific, transferable, efficient, precise, affordable, quick. You can multiplex, that is, do many things at once in terms of editing, but there are still gaps and needs that Greg had mentioned, off-target cleavage, various efficiencies, and I'll mention some of those. In terms of applications, just to be aware, it's not only medicine, but there's a tremendous amount of work being done in the agriculture area, in biotech and commercial industrial applications, as well as medicine. So a lot of resources available for you to look at. And I think we can provide, if people are interested, some references. You already have in your packages some of these, but things about gene therapy basics, you've heard a little bit about that from Greg, studied for more than 40 years, gene therapy, cell therapy, and gene editing, which I will spend much of my time here talking about. And virtually all of that is based, even gene therapy and cell therapy, their fundamental basis is gene editing, and then developing a treatment, as Nicola certainly mentioned. So Greg has introduced ex vivo, ex being outside, vivo life, so outside the body. So cells are removed from the patient, whatever CRISPR-Cas9 in this cartoon delivered to the cells in culture, resulting in the appropriate edit and then reintroduced, or directly in vivo, where CRISPR-Cas9 therapeutic or whatever the methodology used is packaged in a delivery vehicle and then done outside, directly inside the body. So again, Greg has gone through this, the molecular scissors here, which use an RNA to recognize the region of interest. And with this, we can either disrupt a gene, we can correct a gene, or we can regulate the function of the gene, turn its expression up or down or completely off. So everything is really based on this kind of gene editing, and CRISPR-Cas is what's most used now, but certainly the other things, the zinc fingers and talons that Greg talked about, have their function. So gene and cell therapy, gene therapy in the broader sense is the introduction, removal, or change in the content of a person's genetic code. So it all gets back to genome editing with the goal of treating or curing a disease. Transferred genetic material changes how a single protein or group of proteins is produced, and it can be used in the various ways. And Greg has introduced you to all of this. Cell therapy is the transfer of intact live cells into a patient to help lessen or cure a disease. The cells may originate from the patient, which is what we call autologous cells, the patient's own, or a donor, which is referred to as allogenic. And the cells used in cell therapy can be classified by their potential to transform into different cell types. Pluripotent cells, pluri, everything can transform into any cell type, and multipotent can transform into other cell types, but a more limited repertoire than pluripotent cells. And so here, this is just a cartoon of what I just said, so I won't go through this again. These are from the website of the American Society of Gene and Cell Therapy. So if you're interested in looking at this in more detail, you can go to their website, just Google American Society of Gene and Cell Therapy, and you can look at these infographics and things on your own. You're all aware from what we're going through now with COVID of the various things in clinical trials, from preclinical studies to an application period. Phase 1, few participants to look at safety. Phase 2, more participants. And then phase 2, which is many, many more participants, and to look at the efficacy of a therapy. And in order to be approved, all therapeutics must go through this. We hear about it every day and 10 times a day, I'm sure, in terms of COVID things, the vaccines, same applies to therapeutics. So here again at the 30,000 foot level, you've heard about sickle cell disease. This is a very new description of an early phase, first in human trial to correct sickle cell mutation in blood stem cells that cause the disease. And Greg has certainly mentioned this. This is a description involving one of the people, as you see here, Jennifer Doudna, who, as most of you are aware, was one of the pioneers of the development of the CRISPR technology and a co-winner of the Nobel Prize in physiology or medicine, the Nobel Prize this year. You've heard from Nicola about editing for Duchenne's muscular dystrophy, and this is from a recent publication in a journal called Gene Therapy in 2021. So what I'm doing, you've heard about these before, but what I'm doing is providing you some of the updated things that are ongoing now, and there's certainly more in the pipeline. But again, removing cells from the patient, going through the gene editing, and then reintroducing. The FDA approved the first cell-based gene therapy for multiple myeloma. And this is something that many of you who have heard of cancer immunotherapy, this isn't the only cancer immunotherapy, but it's one of what one refers to when one talks about cancer immunotherapy, and that is referred to as CAR T. And we can talk about this later if anybody is interested in it, but it's a cell therapy. And then this was also mentioned, spinal muscular atrophy, which is a very serious condition. One of the ways to treat that, the gene involved is what's called SMN1. And if there isn't enough, one suffers from the disease. One can do things biochemically with gene editing. It's not so much CRISPR-Cas with this. It's involved with a biochemical change in the way proteins are made from messenger RNA. It involves the messenger RNA processing step, and it allows a backup gene called SMN2 to take over, and there's an FDA-approved treatment. Unfortunately, extremely expensive, but it helps counter the effects of SMA. And also, this is from a very recent publication of inherited retinal diseases, where I've had it in bold down here. CRISPR-Cas can permanently and precisely replace or remove genetic mutations. One of the reasons I want to show this is that Greg talked about ways to target things to the right place, to the liver, to whatever, and this can be done biologically. It can also be done physically. And this is one of the reasons why age-dependent macular degeneration, which is what this is about, was chosen. And although many of us may be squeamish about this, this can be injected right into the eye. So you don't have to worry about biological ways of getting it to the right place. And there are lots of other examples of this. So harm, safety, and efficacy, off target, that is hitting the wrong gene, could inactivate essential genes, activate the wrong things like cancer-causing genes, cause rearrangements, or there could be on target, that is going where it's supposed to go, but unintended consequences and changes to the DNA in some cells, but not all cells, which leads to a mixture, which is called a mosaic, and you can change immune responses. So what are the benefits? And you'll hear about this in the next two talks in this session, but in somatic cells, what are somatic cells? They're the main cells of the body. And if you make a change there, it stays in that one person. The analogy I like to use is it's like putting a cast on a broken bone. When the bone is healed, your offspring don't know that you ever had a broken bone. So you can treat blood disorders, lung diseases, essentially. There can be enhancements, that is genes to improve individuals. And this is by virtually everybody in the ethics community, certainly not something that is, well, not only is it not encouraged, it's forbidden in most of the regulations about gene therapy. Germline is used in a research sense to better understand gene function in early embryos, and the germ cells or changes can then be passed on to future generation. And this is where a great deal of ethical thought is being put into. You could change faulty genes that cause devastating diseases and remove them from future generations. There can also be enhancements, which again, I've said is not approved. So clinical use, somatic cells, they're intended to affect only the individual. They can be appropriately and rigorously evaluated, and there are regulatory frameworks, which you will hear more about. Germline, it would be irresponsible at this time to proceed with any clinical use of germline editing unless and until the relevant safety and efficacy issues have been resolved based on appropriate understanding and balancing of risks. Again, you'll hear the next two speakers talk more about this and about the broad societal consequences of this. And this will certainly be a topic that we can discuss in more detail. Most of you have heard about this report from a Chinese researcher almost 3 years ago now about live birth of twins with germline editing. Because of time, I won't say anything more about this, but I hope I can provide significantly more insight and detail about this in the discussion. But I do want to just point out this, that germline editing, is going on. And this is from the Dean of the Faculty of Medicine at Harvard. And if you just look at the last paragraph here, at the conference where the Chinese doctor presented his results, astounding some listeners, the Harvard doctor and stem cell researcher didn't condemn. but instead characterized the Chinese actions as a wrong turn on the right path. The fact that it is possible that the first instance of human germline editing came forward as a misstep should in no way lead us to stick our heads in the sand, Daly said. It's time to start outlining what an actual pathway for clinical translation would be. So there's a lot of controversy about this. The science of the Chinese experiment is very controversial, and I will say right here, the science isn't very good, and we can talk about that a little later. So what is possible? Basic laboratory research, somatic genome editing for diseases, germline editing, genetic changes that can be inherited by future generations, permit clinical trials only for compelling purposes, such as treatment and prevention of serious disease or disability. Human enhancement is not allowed. So Ian Stedman will talk more about this, the role of scientists in policy and politics. So the data comes from the scientist, but the scientist contributes to the reservoir of knowledge, but the decisions must be made by policymakers. So I'll finish just briefly with a couple of comments about personalized medicine, because everybody now, I think, is aware of this terminology. It's probably better called precision medicine. And one of the things that it allows is the right drug to the right patient for the right disease at the right time with the right dose. So there are lots of ways you can think about personalized medicine and lots of research going on. But one of the areas that it is most productive now is in what is called pharmacogenetics. So personal genomics that is targeted to the individual. And the science that we know now allows this, but just focusing on pharmacogenetics, drugs, the right dose to the right person at the right time, you have a group with the same diagnosis and the same prescription. And all of these categories can be treated in a personalized way. So here, the drug is not toxic, but not beneficial. The drug is toxic, but beneficial, and other things as well. And these things can be targeted to the correct individual now when we use personalized medicine, and again, better called precision medicine. So I'll stop here. You'll hear more about some of the ethics and things that I've introduced there. I could, if anybody is interested, talk a little more about some things about personalized medicine and a few very, very recent things in this, if there is interest in the discussion. And I do hope there is time in the discussion to talk more about the science and the ethics of germline gene editing, and particularly the Chinese experiment. So thank you very much.
Thank you so much, Ron. That was a brilliant and very, very fast movement through a whole lot of issues. And I love the fact that you also brought it back into the broader space of, as you say, pharmacogenetics, and also in terms of personalized or precision medicines beyond just the rare diseases. So this is, as you say, a huge area. And I think as we're all appreciating, certainly a lot more science than I think many of us realize, and certainly a lot more controversy as we're beginning to see as well. So it's my real privilege to introduce Ian Stedman. Ian is a former board member of CORD. He's a parent and he's a patient. He's a lawyer. He is a professor in public law and governance, as you see from New York. I'm going to turn it over to Ian, who will take us through some of the ethical and legal issues.
Thanks, Durhan. Hi, everyone. Just a quick thank you to Durhan and to CORD and to Rare Disease International and to RDI for putting on this event. Oh, and for Pfizer, pardon me, and for inviting me to contribute. I very much appreciate that. So as Durhan mentioned, I am a person who has a rare disease. My rare disease is caused by a single gene mutation. The disease is called Muckle-Wells syndrome for those keeping tabs at home. So it's a single gene mutation that could have killed me by now if I hadn't stumbled my way into a diagnosis when I was actually in my early 30s. So because of my diagnosis, I now have access to an effective but very expensive biologic therapy medicine. And I have to take an injection in my subcutaneous, so my fat, every 8 weeks, right? And I have a little girl who's now 8 years old. And because I have a mutation that is what we call autosomal dominant, right? So which means that only one parent has to have that mutation for it to be passed along to the child. So because it's autosomal dominant, I have one of my 3 daughters actually she has the same diagnosis and requires the same medicine every 8 weeks. So believe it or not, I know you've all understand rare disease as well, but we're among the lucky ones. 95% of people with rare diseases don't have access to an effective therapy. And I tell you this, I remind you of this because I'm going to talk to you today about the ethics and the law, to some extent, of genome editing. But I myself could in many ways personally benefit from this technology. At least I think I could, not knowing the science as well as everyone else, but I believe that. And you know now that I have a daughter who knows, maybe wants to have kids of her own one day. And so I can imagine that this technology may be of interest to her one day too. Not just somatic, not just in her body, but maybe also germline, so that this disease doesn't get passed on and she doesn't have that risk of, 50-50 her children getting it. I don't know, but I'm pretty sure that's going to be an interesting conversation to have with her when she's old enough to have it. So with that said, let's talk about some of the more common concerns that have been expressed in the literature about the possibility of editing the human genome. We're going to bounce back and forth a bit between the somatic and germline editing, because many of the concerns about somatic editing apply equally to germline, but there are also going to be special concerns that arise in the context of germline editing. And we'll do our best to make sure those things are clear. I see in the chat. Oh, okay. Not about me. Good. So we'll make sure that those differences are clear when they come up. So let's start our discussion from the assumption that both types of genome editing in humans are possible. So we're kind of forward thinking a little bit here. We know that somatic is exciting and happening very actively, and that we have this one example in China of the germline editing. But let's just start with the assumption that this is all going to be possible if it isn't already. So then, if we do allow genome editing, when should we allow it? If we are too permissive, and if we allow this technology to be used for anything that it can possibly be used for, then one of the criticisms of an approach like that in the literature that comes out is that we risk pathologizing our differences. So in other words, we risk implying by our actions that when a person is different, or that when they will be born with something about themselves that will be different from, say, maybe what their parents expect, or what is average or normal, or, then if we're too permissive with this technology, maybe we risk sending this signal that they need to be fixed. So let me unpack that a bit. So this language of pathologizing means that our differences would need to be fixed. Our differences, our illnesses, our sickness would need to be fixed by modern medicine, because all difference is bad in some way, and we shouldn't allow for it. So taking this kind of mentality towards the use of this technology is obviously problematic, I think. It looks at disease and disability through a lens that would minimize the value of those who live with these differences. And it doesn't acknowledge how what often makes living with disease and disability challenging is that we have set up our societies in ways that exclude or alienate or minimize support for those who need a little extra something. So, a little extra accommodation in order to be treated equitably, in order to have equitable access. So if we pathologize difference, then we kind of ignore the social aspects that create the challenge that difference brings. If we do that, that is, if we aren't careful about not being too permissive with the technology, then the argument has been made that we risk perhaps a culture shift over time. So maybe this means we might slowly slip into a society where the pathologizing difference, pardon me, I'm not going to try to say the word in a larger way, pathologizing our difference leads to a type of stigma or pressure to edit those certain gene variants when they're found or when they're potentially to be found. So now let me use myself an example to try to put this into a more grounded context. Imagine that I knew my child had a genetic mutation before she was born. Right, after all, it's a 50-50 chance. And it is possible now to test embryos for the mutation that I know I have. So we could test an embryo before we implanted it, for example. So imagine I know that unless I edit my future child's genome before she's born, then she's going to need $100,000 or more of medicine every year for the rest of her life. So, and this isn't a far-fetched example. Imagine I live in a society that has pharmacare. We're hoping Canada at some point does. And imagine pharmacare, the government, covers 100% of that $100,000 of medicine every year. But the government also covers genome editing. And so imagine again that genome editing is advanced enough that we can do this safely, and that it costs way less in the long run to do genome editing up front or in an embryo than to actually pay for medicine over the course of a child's life. So you can see that there's a possible world ahead of us, if we take all these things as possible, that puts me in a position where government policies may subtly or even not so subtly pressure me and my family towards editing an embryo of our future child in order to save costs for a healthcare system that we all know is perpetually complaining about being stretched. No matter which country we live in, our healthcare systems are always perpetually complaining about being stretched for dollars. So this kind of scenario might have an impact on parents like me. If it becomes cheaper to edit than to treat over the long run, there's no cure for what I have, there is treatment. But if it becomes cheaper to cure it by editing it, will parents like me look ahead of them at the future of their children and see expenses everywhere and see costs everywhere and see the burdens that I'm placing on others and on the healthcare system, right? So will I start to think about my child's future in a way that makes me think more liberally about the need for gene editing? And do we as a society want that? Do our societies value difference? If we do, then we have to be careful not to push ourselves towards this kind of pathway where we try to pathologize difference and treat it and cure it and consider it something that needs to be eliminated up front, for example. So should we be concerned about all that, and should we do something to protect against the potential for what many have called in the literature the possibility of designer babies? So Ron mentioned it as enhancement. Think of using gene editing for enhancement as a kind of approach towards making designer babies. If you can identify the gene, you could edit the gene. And who knows what the genes control? Maybe eye color, maybe hair color, maybe the musculature. Okay. So before we start to think about law and policy options, it's easy to see that it all gets pretty complicated pretty quickly. But let's pause there, because so far everything I've said could really be about somatic cell editing, right? But if we are editing the germline, then we have another huge problem. And that is, how do we even figure out whether that is possible? How do we design experiments to test germline genome editing when we know that this will require us to monitor safety and efficacy in not just one generation, but in future generations? Are we willing to just assume that we've got it right, like it happened in China? Or do we need someone to decide to have their germline edited and then to decide to have children, so that we can then test the children and see if that edit was passed along safely? How many generations do we want to experiment on? And how do we do that ethically when we really aren't going to get their consent to be born, to be part of our experiment? So these are the kinds of questions that come up. The last two things I want to mention here are that we already have massive health equity issues in countries like Canada and the United States, and really everywhere in the world. We know that people who are racialized and people who are poor have a much worse access to healthcare than, generally speaking, do the white and the affluent. So if this technology becomes viable, we should want it to benefit everyone, as I think we should want all technologies. So we need to ask ourselves questions about who gets access to genome editing and why. Is this a technology that we think should be commercialized so that some company can profit off of it? Or is it the kind of technology that we should want available at a low cost for broad public health purposes? And the answer is, I think it depends. But we need to figure out what these questions are, and we need to put these potential concerns on the table so that we're not blind to them as we move forward with the science. In my next slide, I want to identify some legal issues. The first one, I'm speaking a little quicker, Durhan told me to hurry up. The first one stems directly from the last ethical issue and is really about commercialization versus low-cost access for public health. Governments make decisions about things in part by deciding whether or not to allow inventors, who are usually maybe companies, right, to claim and register intellectual property rights. So having intellectual property rights means that you have the exclusive right to control or to exclude those who use the technology. So you decide who gets to use your technology if it's registered to you and for what purposes. So those rights are, they last depending on the regime and where you live for some predefined period of time. So the question then is, should gene editing technologies that have this broad potential for public health benefit, should they be protectable by intellectual property or not? And when and why? Those are important questions that lawmakers will have to consider. Maybe it depends on who uses it and for what. And if we're talking about healthcare, then I think we can all agree that some regulatory oversight should be in place to ensure the technology is safe, right? But I just want to mention briefly that the technology will be used in areas outside of healthcare as well, and that there will need to be other oversight and regulatory interventions. In Canada, we have something called the Genetic Non-Discrimination Act. And so this is a piece of legislation, very, very short, that prevents insurers and employers from discriminating against someone like me because they know I have a genetic disease, for example, that would cause them to have to spend money on medicine for me every month. So we have a limitation on discriminating on the basis of genetics. So one can imagine a world in which stigma becomes attached to parents for not correcting what they should know are pathogenic genetic variants in their children. And so maybe our human rights laws or anti-discrimination laws need to step in and play some role as well. It's impossible to really know what to expect unless and until we see the technology really in full action. Right now, we're just kind of imagining a world, and I think we all believe in the literature, at least takes this position, that we need to remain flexible and ready to adapt so that laws can change and regulations can adapt in order to protect people. Okay, so move on quick. In Canada, we currently have a criminal ban against making alterations to the genome. And you'll see here, this is the language of it. So alterations to the genome that are capable of being transmitted to descendants. This is a ban in our criminal law that exists from 2004. It clearly applies to hereditable genome editing. But although in 2004, that really wasn't a technology that was in the public consciousness, there are other countries around the world who have used the criminal law as well to ban germline genome editing. We can come back to that in the discussion. I'll move through it quickly now. So where does this leave us? It leaves us with a whole bunch of policy questions that we need to consider. Do we really as a society think that germline editing is acceptable? Maybe not yet. If so, when will we? And what level of risk will we be able to tolerate for maybe unintended side effects? And what kind of therapeutic purposes are we going to want to use it for? How do we prevent the improper use of technologies like this that could, in the wrong hands, do some real damage? Right? Is it something serious enough that we want to use the criminal law for? Or is it something that we should be developing greater social architecture for? Meaning, should we be teaching more about it so that we have it as part of our curriculum so that we can create a social architecture that incentivizes the behaviors we'd like to see, which is avoiding improper uses, right? And if we do allow genome editing in humans, how are we actually going to design these multi-generational clinical trials that we talked about could be problematic earlier? The only way to figure all these things out is to keep on top of it and to do our best to get everyone around the table, everyone who has an interest in genome editing, whether directly or indirectly. We need patients and their caregivers to understand the technology. We need to identify the issues and talk about them. We need scientists and social scientists to help us unpack how the technology works and what its implications are, healthcare providers, and we even like faith leaders, absolutely, because I don't like the expression playing God, but many people believe that a technology like this is unnatural in some ways, and so we need to hear their voices and take them seriously. And we also finally need special interest groups, industry, research funders, and all of you who are here, who may not fit into these categories, but have an interest, to sit around the table and to learn about the technology so we can have nuanced conversations before we get too far down the road. I'll stop there. Thank you.
Thank you so much, Ian. Ian always does this to me. I always start off so optimistic with him in terms of, hearing his personal stories and the good news. And by the time I get done, it's sort of like, oh my God, this is so depressing. This is so challenging. So thanks a lot, Ian. You never fail to disappoint me in terms of putting it back into the quagmire at the end of it. So hopefully we're going to get Nick Mead, who is with Genetic Alliance UK, who's going to dig us back out of that hole. Nick, thank you so much for joining us. Nick is one of the long-time sort of lay public patient experts in this area, and we're so delighted that he was able to make the time to join us. So Nick, I'm going to turn it over to you.
Thanks very much for the complimentary introduction there, Dirhan, and thanks very much for inviting me today. It's a pleasure to be part of this meeting and to follow on from Ron and Ian. Hopefully what I've got to say builds on the points they've already made in the discussion today. So I'm just going to reflect today on the work that Genetic Alliance UK has done on genome editing, really to understand our own community's views, and then we spend a lot of time then trying to transmit these views to policymakers. So I'd like to acknowledge all of the people within our community that have made me able to give this presentation by sharing their perspectives and working with us in workshops, and also Eurodis, who I'm sure you'll be familiar with. And Progress Educational Trust is a UK organisation that focuses on issues relating to genetics and ethics in the UK. So thank you to them. Going to start just with a very quick introduction of our first report that we did on genome editing, which is quite old now, but hopefully still reflects the community's views in the UK. We delivered a series of educational webinars to bring our community up to speed on the topic. And then we asked them to fill in surveys and report back their views on genome editing. The first clear point to make here is that our community welcomes the use of genome editing technologies in the research and clinical setting to develop cures and treatments for unmet health need. That there's a strong level of interest within our community to learn more. And it's interesting to note this point came after a educational session. So curiosity is high, and we need to keep on doing events like these. The community came back with a clear message that use in humans should be limited to treating medical conditions. And this is a point that comes up in ethical discussions sometimes around enhancement versus treatment. And our community is clear that we need to focus on medical uses to treat unmet health needs before we start looking at how we can make healthy humans better. We, as a community, believe that the potential benefits of this new and innovative technology far outweigh potential risks, and therefore there's a duty to investigate the potential for genome editing to improve the lives of people living with rare and genetic conditions. And the last point here is that we think the patient voice is an important one around regulatory decisions for future access to genome editing, and we need to keep around the table making sure that our views are heard there. And there's a link here to the full report. I'm hoping that my slides can be shared after this meeting, where you can see that report that was part of a European project called NERI. So then I'm going to move on now to our second report, which we did in the following year. And this was a collaboration with the Progress Educational Trust. This is a bit of a different approach. We brought together 18 patients and carers to, so also people from the fertility community as well, to have a series of 5-day-long workshops. And the output of this was actually around clarity of communication about genome editing, and hopefully I've kept to my own rules and always use the term genome editing today. That was one of our recommendations, to really be clear, and there's the link to that report's here. So there's some reasons why we thought this was necessary. We think definition is crucial to progress understanding and work towards common ground, and that's an experience that we've seen in sometimes stalling progress towards understanding of big data, personalized medicine, and another controversial reproductive technology that has been approved for use in the UK, which is called mitochondrial replacement therapy, sometimes called 3-parent babies. But in all of these, it's our strong view that a clear definition of what we were talking about would have helped patient engagement in the dialogue a lot earlier. And genome editing is a very broad topic that has uses, many uses within humans, germline or somatics use, and then outside of health could be used for enhancement, and then outside of human use could be used for crops, for in insects to fight disease, and in lots of other areas as well that I know very little about. So it's very, very important for me to categorize what I'm talking about, and you know, what the message is from us, and where it lands. That report is there for you to look at as well. Now, the bottom line from the work that we've done on this is that we're strongly in support of genome editing as a technology to potentially improve or address unmet health need. One of the best quotes from all of the work that we did was that we feel that any person with a life-changing or life-limiting condition should be given the chance of a normal life. We expect that there's use here in research, in understanding conditions, and also eventually we think that some of the reproductive choice options need to be understood as well. I won't go into detail on some of these points because I know we could have some discussion. I'll just end with hopefully, and I think this links back to some of Ian's statements, who should have the primary viewpoint on our future relationship with genome editing, and we would say that, you know, who stands to benefit most from progress? It's the patient community, and as we trial genome editing, it will be our patient community that are taking risks as we use them in people for the first time. So we would say that patients, carers, and families with unmet health needs are the ultimate stakeholder in this debate, and their experience should frame discussion as we go forward. So thanks very much.
That was brilliant. And thank you so much, Nick, for making, I think, available the discussions that have taken place. And as you say, 2016, 2017, but those are still the same issues that we're dealing with, except that we've got a lot more advancements in science now, and we've got a lot more practical applications. So it makes it, you know, so much more challenging. We're going to go into breakout sessions and have the opportunity for everybody to do a deeper dive within their own sessions in this, and really encourage everybody to speak up, you know, strongly. But maybe before Nick and Ron and Ian and Greg go into the breakout sessions, though I know you can't stay, Nick, maybe I'll just get this last panel, get each one of you to make a comment based on kind of where you sit now. And based on the issues as you've heard them, you know, kind of raised here amongst others, to what degree do you think patients have the right to make those ultimate decisions. And what about, as Ian says, not even just from a public health financing point of view, but just from the public point of view, what should be what's the balance between what the public belief should be and whether there is, we know that we can today do some of the things around selective not selective genetics, but we, in many countries, right, we do the testing to see if a child might, be carrying a risk in terms of Down syndrome, and we may make some decisions based on that. We can do some early genetic pre-screening. What's the right of the patient versus the right versus the public in terms of these decisions? Is it always just a patient decision? Is it something that goes beyond that?
I'll
Ian.
get started. So I think it all starts with the science. I don't think we get to the patients having a real discretionary right to anything until the science is effective, safe, and established. But then the next question is, if it's established, but we do also know that there is some level of risk, I think that's the point at which we have to say to patients, educate the patient and let them make autonomous decisions, right? Let them make a choice about what level of risk they're willing to assume for the potential for a therapy using these technologies. But I think the starting point always has to be that the science is there first before we start trying to figure out what the risks are and asking the patient to exercise their discretion. That would be my contribution. See what the others think.
Yeah, Nick or Ron?
I think I just I agree with Ian. I think as far as the public perspective on genome editing is and goes and develops, that we have to understand that that's a naive, casual perspective. If you're a member of the public, you've got no major reason to strongly engage with genome editing other than it's an interesting, new and novel thing. So we need to give them the reason to care about it. And I hope my last slide points out that the reason is that there's a significant amount of unmet health need at the moment. We've got, a few more than 200 medicines for rare conditions and more than 6,000 rare conditions. So we see genome editing as one of many possible avenues to develop cures and treatments for unmet health need. And we need to communicate to the public how important to make that progress is. And hopefully then they will see genome editing in that light and care about it and hopefully agree with us.
So Ron, where would you weigh it in on this? If you're speaking, yeah.
That's why. Yeah, I can just, I echo what both Nick and Ian said, that it's got to be science-based, but the community or society, we've got to do, and that includes us as scientists, have to do a much better job of outreach, science outreach, what I call science literacy. And so it's sort of education, but not everybody can understand and can't be expected to understand the details and the nuances, even at the fairly lay level that we've heard this morning about all of the science. But what is important is to understand how science is done, why these questions are important, how things move forward, and step by step and over long periods of time based on a solid fundamentals. And so that's what's really important, is science literacy and developing what I like to call a science literate society, which doesn't mean understanding the details and the nuances of every technique and technology, but understand science in its broadest sense. So then the informed decisions can be made by the stakeholders, and clearly the patients are at the top of a stakeholder list.
Thanks a lot. So this is why we do these things, right? We need to be an informed community. So you see here, I popped up the scenario. So first of all, huge thanks to Nick, Ron, and Ian for starting us on this again, this phase of what we need to consider here. And again, we could do many hours more of this, so appreciate very much. We're going to move you into breakout groups and hopefully, continue this discussion, obviously, and we're going to try to judiciously place Ron, Ian, Greg, and Nicola into different groups. We are going to ask you, this is just a scenario. You don't have to stick to this scenario. It's just to put in context everything you've heard. So what you'll see is that the scenario is a rare inherited disease. Those of you will see that it's loosely Huntington's disease, but it's not tied to it exactly, but it is pretty much that. It is a multi-systemic disease, so it breaks down the brain nerve cells, but it infects then people's physical movements, their thinking, and psychiatric disorders. A very challenging disorder. There are no effective treatments or cures. Symptoms develop at age 30 to 50, which means in many cases, these people do not know that they're symptomatic, and they may actually be carrying this gene until they're actually have already had children, for example. But the time to death then is some 10 to 30 years afterwards. It is a single gene defect, so we've heard that already. It is autosomal dominant, which is what Ian talked about. That means you only have to have one copy of it for you to have the symptoms, and there's a 50-50 chance then that your child will actually have a copy of that gene and may also then have symptoms. So there are three options that we're asking you to consider in your small groups, and you just use them as starter points, right? It's everything you've heard about right now. The three approaches are a AAV vector. It will deliver then this microRNA directly to the brain. It will knock out that DH gene, the one that's creating the problems, but the problem is that it will also then reduce the normal protein that it produces. So the trouble is that we don't know what the effect that is to totally knock out the gene. The second option is a genome editing using maybe a CRISPR-Cas9 technique. You introduce a tool, you cut out the defective portion of the gene, you bring it down to normal size. It's a bit of what Nicola was talking about. The gene is actually just has too many copies in it. So these copies keep making too much protein. You cut out the excessive portions. Hopefully one of the things that happens is that it now acts like a normal gene. The challenge is that we got to get it to exactly the right spot, not cut out too much, not target the wrong cells. There's that risk. The third one is a bit of what Ian's talking about and Nick as well, genome editing. We could do it in the embryo. If you know you're at risk, you can extract the embryo ahead of time. You may in fact be able to edit that embryo before you actually reimplant it. You could do it in the fetus as well, but we're not really thinking of that part. So there's no possibility, but because you've actually edited in the embryo, you also know that any offspring of this child who's born with this altered genome is not going to pass it on to anybody else. But we don't know what the consequences are of actually editing. There are other mutations, as Ron mentioned, there are other risks. So we're going to throw you into these shops. We don't have a whole lot of time, unfortunately, but we do hope that you can have a fairly lively discussion within your workshops. Use this just as a starter point. You may, in fact, discuss it as you wish. We're going to ask that there, hopefully one of the four leaders that we've had speak so far will be in each one of the groups, but we're going to ask somebody to also be able to help lead the discussion, take notes, and we're going to have a brief report back when we get done. So you have about 15 minutes. You're going to move right now. very, very, very tiny bit of time left. I think we'll just do a really quick go around. I see Greg on my screen, so I might just ask you, Greg, to kind of give us a quick shout back in terms of what happened in your group.
Well, we talked about, one theme was the durability of these treatments, so how long will they last? So there was the one treatment talked about AAV and the microRNA. And so there were questions about how long that would last, with worries that it wouldn't, persist. And then on the other end, thinking about the germline editing, there was, comments about, a lot of those effects could happen way in the future, decades in the future, and future generations, right? So, so there's those extremes of the time effect there that were were one point of discussion. And then, of course, there were discussions about off-target effects as well, like with CRISPR. That middle section posed, the CRISPR could have a benefit, but it could have the side effect as well. And we talked a little bit about the risk-benefit ratio there for the patients. You have to look at the disease, the severity of the disease, the specificity of the genome editing reagents, and then clinicians and scientists and patients all have to get together and sort of make a risk-benefit ratio sort of decision, for each of those. So,
thanks a lot. Maybe Ron, would you like to give us a quick reprise in terms of your group there?
Sure. We didn't get into the nitty-gritty that Greg talked about initially to the extent that it sounds like their group did. A few people talked about the organizations that they were involved with and their own personal things and hopes for the future. We did spend some time talking about, again, what Greg mentioned at the end of what he said, and that's the future. And the fact that for all too long in academic history, that the natural sciences and the social sciences and humanities have been two separate silos and haven't worked together. And now there is in the last 20 or more years, there is a change in that we're talking about health and not medicine only. So we're getting away a little bit from the medicalization of health. It's still very important, and biomedicine, as we're seeing with all these technologies, is the basis of what we're able to do, but we're beginning to get everybody involved, and it's important that they're going to work together at the same pace so the science doesn't run as far ahead as has happened in the past, and then we can't do anything with the science because the ethical questions haven't even been raised, or the legal questions. So we've got to move things together at a similar pace.
Yeah, thanks. That's a great discussion, I'm sure. I mean, I think some people would challenge and say, I think we've already lost that race. I think the science is so far ahead of us in terms of certainly our understanding and the legal and the ethical. That's why these discussions are so important. Ian.
So we also didn't talk about the scenarios, but we had a very good conversation about all the other things and how important this engagement in the topic really is. As we were moving out to the break rooms, Encarnacion made a comment about the importance of education and those who don't have a voice. And so one of the things that we really identified was that it's kind of a chicken and egg thing, right? In the sense that we want the science to come so that we can actually be confident and really engage in conversations about its potential. But we also want the public to be aware of the potential of the science, because it's when the public is aware that they can pressure the policymakers and those who spend money on science, right, the public research dollars, to actually invest and prioritize things like this. And if Canada, where I am, we have a public healthcare system, and so we should want more public funding going into this science, just so we can see if the potential is there to really help tackle that 95% maybe of problems of those rare disease patients who don't have therapies. So we talked an awful lot about the importance of a broad engagement. And then a conversation came up about how hard it is to regulate this space. What do our lawmakers do? And I think we kind of fell back to an understanding that it's very hard. And it's very hard to regulate a lot of spaces. Artificial intelligence was another good example. And what we can do is to help raise awareness and create a kind of collective consciousness about this topic so that we can continue the conversation and make it a point of discussion, perhaps in those political times when we're voting people into office, right? So we can ask them to prioritize health.
So where we have a public system, this is something that we can certainly, I think, try to interject more into. Monica, before you go, I'm going to ask you just give us some reflections as well. I know you led off our discussion. Happy to always have you kind of chime back in here.
So thank you, Johan. Our discussion was, I think we were the classic, almost overwhelmed by the scenarios at the start, where it was like, wow, to be in a position to make decisions like this is just so overwhelming for families, for parents, for individuals. And I think we dwelt around our personal experiences in that space, and everyone offered interpretations that kept coming back to it's such a difficult decision to make, and there are so many considerations. And I think at the end we also came back to it's also a really individual decision. So, you know, we've just had that discussion, all the legal and societal and cultural kind of things, but at the end of the day, it's also such an individual decision, and the individual education and the individual consent and understanding is so critical as well.
Yeah, that was so powerful. And basically, I think a lot of people are saying it's at least great that we have the options to be making those kinds of individual decisions. I don't know, Nicola, I'm going to give you the last word because I know we're already over time and we want to properly close off. But Nicola, if you don't mind.
Sure, thanks, Derhan. I'll keep it brief. I think the bottom line here was, one, this scenario was quite overwhelming because we're talking about clinical trial. There's still a lot of unknowns. So I think the first and foremost is eliminating as much of the unknowns as possible. The education and the knowledge is key and paramount to making decisions. And then the last piece is it's very much an individual decision. What I choose versus what someone else's choose, it's going to be very individualized. It's going to be based on the circumstances of that person's condition. It's going to be based on the circumstances of the risk benefits. And it's very, very much an individualized situation. So I don't know what else to say to that other than, you know, it's not easy, but I'm happy to have these opportunities, right?
You can imagine, based on what everybody's saying here, what this landscape is going to look like. If we fast forward to 5 years from now, 10 years from now, what is this landscape? But I think Ron made the important comment. We've got to get the social, the legal, the ethical to stay apace with it. Otherwise, we will find ourselves with amazing, you know, kind of options, but we're not going to be able to actually appropriately be able to be engaged in that decision making. As you can see, the science is not easy. I'm always impressed when I hear Nicola speak, because I know you come from certainly a science background, but you're not a deep scientist in this area, and yet your command of the information is so amazing. So it's always a thrill to hear you kind of restate some of the things for all of us in language that we can all appreciate, knowing that you do that great bridge. And this is what I'm hoping all of you will become. You will become that bridge to helping us make sure that other people in our community are informed. It is 5 over. I apologize for everybody. I hate to keep people hanging. I do thank you so much for the participation. Thanks to our panelists for their amazing contributions. We'll send you out a brief survey. Hopefully you'll have a chance to be able to respond to it. All the materials here will be available on in your folder, so be sure to look for them. If you've got any other questions, I'm sure if you want to send them in, we can get any one of our speakers or others to respond to it, and hopefully we can develop an ongoing community here. Our next session will not be until June, but it will be equally exciting. We're going to look at some of the CAR-T cellular therapies, look at the lentivirals in terms of their application, and we're actually going to hopefully have a very important discussion around equity and access. What are we going to do about making these therapies available across the globe? Because obviously they are expensive. Obviously they require a lot of investment, but you know, obviously they could have a huge amount of benefit beyond that. So I think it's hugely important that we enter that discussion. So we'll have a very deliberate discussion on it, and maybe, you know, beyond even these 3 sessions. So thank you folks so much for your continued participation. And we look forward to seeing you in our follow-up sessions. I'm sure I've forgotten a lot in terms of closing this off. I always do, but be sure and write to us, and huge thanks to our collaboration with RDI for making this happen. Thanks a lot, folks. Bye-bye.
Thank you. Bye, everyone.
Bye.
Bye, thank you.
Bye.
Thank
Bye.
you.